So good morning, everybody, and you're very welcome to this morning's Institute of Food and Health Research Bites. So this morning we're delighted to welcome Dr. Martina Wallace, and we're in some ways also welcoming her to UCD because she joined us in Ad Astra in May, but we haven't had the opportunity to really welcome you face to face, Martina. So we're delighted to have you on board, and thank you so much for doing this research bite this morning. So although Martina is just back with us recently, she did start out in UCD with her degree in biochemistry and her PhD and a postdoc subsequently in the area of metabolomics in IVF, and then moving on to diabetes and obesity. And then she took a trip abroad in 2013, traveling to San Diego in working at the University of California in the bioengineering department. And there she furthered her work and developed her expertise in metabolic systems biology. So she's now busy setting up her own lab, and this is going to focus on metabolic dysregulation and diseases associated with the metabolic syndrome. So over to you, Martina, if you want to begin.
All right, thanks very much, Dolores. I'll just share this now. So I think that's clear. Okay, so thanks very much for the introduction. So today I'm going to talk about tracing new insights into nutrient metabolism, which is basically about how I've used isotope tracers to kind of gain new insight into how, or how we can use them to gain new insight into how nutrients are metabolized under different physiological contexts. So as Dolores mentioned, for both my PhD, which I did here in UCD, and then also my subsequent postdoc in UCSD, most of my research has focused on metabolic dysregulation in disease. So my main focus for the last few years has been on diabetes and obesity, but I've also worked on things like macular disease, infertility, and cancer. And then here in UCD, I'm really hoping to focus on the relationship between diet and metabolic pathways in lipogenic tissues, so tissues such as the liver and adipose tissue. And I want to understand how nutrients are metabolized under different physiological contexts. So things like the inflammatory state of a tissue, or also oxygen tension due to differences in vascularization, can all change how our tissues metabolize nutrients and what pathways they direct them down. And so then this leads to different intermediates of metabolism and different products of metabolism, and this can all have different impacts on cell function. So my main questions then are the factors that drive altered nutrient metabolism, and what exactly are they, and how this impacts cell function. Okay, so then what are the main challenges in metabolic research and kind of understanding how nutrients are metabolized? Well, the first kind of main challenge is that metabolism is really highly interconnected and can have a lot of redundancy and flexibility in many enzymes. So this is kind of the classic metabolic pathway that's shown to, that's used to demonstrate the interconnectivity of metabolism. And you can see it's a real complex network, so you have to look at metabolism on a network level. And then finally, what I mean by redundancy and flexibility in enzymes is that there's a lot of isozymes. So if you knock out one enzyme, oftentimes another enzyme can easily compensate, or there can be lots of non-canonical products of enzyme activity. So a lot of these metabolic enzymes can do side reactions or have use alternate substrates. And so when you knock out an enzyme or alter an enzyme in metabolism, you can also be altering alternate functions that you don't know about, so, or that we don't fully understand yet. The second main challenge is that genes and protein levels don't always correlate with pathway activity. So although looking at altered protein levels can give you an indication that specific metabolic pathways are altered with disease, oftentimes flux can be more modulated by things like post-translational modification of those enzymes or substrate product ratios. So it's really important to directly measure metabolic pathway activity, and protein and gene levels don't always correlate with that activity. And then finally, one of the biggest challenges is the subcompartmentalization of metabolism. So for example, when we look at metabolites in a tissue, there's a lot of different cell types obviously in most tissues, so you don't know which cell type is contributing most to, say, altered metabolism. Or when you look at a single cell, there can be big differences in the metabolite concentration between things like the mitochondria and cytosol. And so understanding how metabolism is changed, we can be challenged because we don't understand the differences in those metabolite concentrations between those two compartments, or it's really hard to capture those differences in metabolism between different compartments. So because of these challenges, there's a range of different approaches that are commonly used to study metabolism. So the classic way is, the classic kind of biochemical way is isolated enzyme kinetics. So this is really good because you can get detailed knowledge on one enzyme, but it lacks the benefit of studying how reactions perform in a network. So the next main way, or the most common way that people use now, is looking at metabolite levels, or metabolomics. So you get this really broad fingerprint of how metabolite levels change with disease, or between a certain environment, or how it changes with certain environmental factors. And this is really good for generating hypotheses, and it's also really well suited to patient work, but it has limitations. So for example, you could have an increase in metabolite C, but you don't know if that's because of a decrease in activity of enzyme Y or an increase in activity of enzyme X. So it's really hard to identify the exact node of regulation. And then in addition to that, metabolite levels don't always correlate with flux. So you can have high metabolite levels across a specific pathway, but that doesn't mean that there's higher flux through that pathway compared to a different metabolite pathway. And then the approach I've been for the last couple of years, in combination with more classic metabolomics, is using isotope tracers and calculating metabolic fluxes. So this has become really popular in the last 10 years, but it's been around for much longer than that. And basically what normally happens is you could feed like a 13C labeled or heavy labeled glucose or amino acid. That's then metabolized by the cells, and that heavy label is incorporated into downstream metabolic intermediates. And then by looking at the enrichment of those downstream metabolic intermediates, you're able to determine fluxes. You're also able to get more information on directionality. You're able to uncover new pathway architecture, so by following where that carbon goes, sometimes to unexpected places. And you're also able to get an idea of relative substrate utilization, so whether or not tissues like to change what substrates or nutrients they use under different environmental factors. Okay, so then the typical setup for an experiment like this is we would use heavy labeled glucose or amino acids, so they can be 13C or deuterium labeled. Feed these to cells or mice or to humans, and then we look at incorporation of that heavy label into downstream metabolites using things like GCMS or LCMS. And then from that, we can quantitate changes in flux or look at differences in substrate utilization. And then we can identify pathways and mechanisms. So by combining with molecular biology approaches, say to knock out enzymes or overexpress, we can dig deeper into mechanisms and then identify nodes of metabolism that would either be good targets for dietary intervention or for drug targets. And so today, I'm just going to kind of briefly give an overview of my work on branched-chain amino acid metabolism. This is a good example of using isotope tracers to understand how amino acid metabolism can change under different contexts. So branched-chain amino acids and their altered metabolism is associated with a number of different diseases. So they're commonly elevated in patients with cardiovascular disease and insulin resistance. Alterations in their metabolism is associated with cancer formation and progression. And there's also at least 16 different inborn errors of metabolism associated with the breakdown of these branched amino acids. So it can be quite a long and complex pathway, and mutations in any of the enzymes associated with this all lead to disease. So here on the right, I'm just showing how these branched amino acids are metabolized in the cell and what kind of their main functions in the cell are. So they're essential amino acids, which means they're essential for protein synthesis, and they can also perform signaling roles in the cell where they can signal through things such as mTOR, so they're important for growth. Then, in addition to that, through their metabolism, they're really important nitrogen donors. So the first step of their metabolism, that nitrogen is kicked off and can be used for non-essential amino acid synthesis. So if you disrupt this step, you see you can have detrimental impacts on the synthesis of other non-essential amino acids. And then they can also be broken down and their carbon used to feed the TCA cycle. So they're just important for energetics in the cell and oxidative TCA metabolism. And my work is mainly, or how I started working on this is I was interested in the role of branched amino acids in insulin resistance, as although they're elevated in these patients, it's unknown whether these elevated levels are causative and so can actually drive tissue dysfunction, or it's just correlative with and they're just a biomarker of altered tissue function in these patients. And so to start understanding how branched amino acid metabolism and why it might be important in type 2 diabetes and obesity, we started to look at 3-3L1s, which are a model of adipose tissue in cell culture. And most people at the time, when I started working on this, were focused on muscle, as this is really the main site of branched amino acid catabolism in the body. But there's also now been a lot of evidence showing that the adipose tissue is also an important regulator of branched chain amino acid metabolism. So basically what we did is we took proliferating pre-adipocytes, which are more like fibroblasts, and differentiated them into these lipid-laden adipocytes. And then we looked at how their metabolism changed. And so in this graph here, I'm showing citrate enrichment, so the TCA cycle intermediate, from heavy labeled leucine or isoleucine. And as you can see with differentiation into adipocytes, there's this massive kind of upregulation of branched amino acid catabolism. Then in addition to that, we looked at then all the different sources these cells used for fatty acid synthesis for lipid droplet formation. And what we found was that whereas in pre-adipocytes and actually in most cells in culture, glucose and glutamine are the main source of carbon for fatty acid synthesis. But in adipocytes, only 30% of the carbon for this de novo lipogenesis now comes from branched chain amino acids. And then finally, we wanted to understand, well, was this some kind of artifact of cell culture, or does this also happen in vivo? So what we did is we fed labeled branched amino acids to mice, and then we looked in our adipose tissue to see were they utilizing a lot of branched amino acids, and we found that there was actually similar TCA cycle enrichment between the 3-3L1s, which are shown in gray here, and the white adipose tissue, indicating this wasn't an artifact of cell culture, and branched chain amino acids are an important carbon source for adipose tissue. So then the next question was, well, what are the drivers of altered branched amino acid catabolism in patients with insulin resistance? So there had been some previous work showing that expression of proteins in the branched amino acid catabolic pathway are decreased in the adipose tissue of those patients with obesity or type 2 diabetes. And also when we fed labeled branched amino acids to high-fat diet-fed mice, so a model of obesity, we found there was a general suppression of branched amino acid catabolism across all the different tissues we looked at, including the adipose tissue. And so we wanted to try and identify the main factors that drive this. So we tested a range of different factors that play a role in adipose tissue dysfunction. So we treated the cells with inflammatory cytokines such as TNF-alpha, and we also induced ER stress. And then we also looked at hypoxia, or low oxygen levels, as this has also been implicated in adipose tissue dysfunction. And what we found was that hypoxia was the main driver of this altered branched chain amino acid metabolism. So here I'm showing again the TCA cycle intermediates and their enrichment from branched amino acids. You can see that it's really significantly suppressed in those cells that are exposed to hypoxia, meaning that it's really shutting down the breakdown of these branched chain amino acids. And then we looked in other cell types. So we looked in primary brown adipose tissue, also in primary human myotubes and human white adipocytes. And you see that hypoxia decreased branched chain amino acid catabolism in all of these. But the biggest impact by far was in white adipocytes. And then finally, we also looked at oxygen levels in the adipose tissue of high-fat diet-fed mice. And other people have shown this before, but what we found was that there's a significant decrease in oxygen tension in the adipose tissue of these mice who were on the high-fat diet, where there was no change in oxygen levels in the muscle or liver. So this shows that obesity-induced hypoxia can cause changes in branched amino acid catabolism, and maybe may contribute to the elevated levels found in obese and insulin-resistant patients. Then the other thing that we found was that these branched amino acids can also be used to make branched chain fatty acids, or these monomethyl branched chain fatty acids. So these have been found in humans before, and they were thought to be primarily diet-derived, that they're abundant in dairy products and things like this. But what we found is that actually a significant amount of these we make ourselves by our adipose tissue. And so basically what we found was that fatty acid synthase, which is the main of the enzyme involved in de novo lipogenesis, was using these intermediates of branched chain amino acid catabolism to start an acyl chain and then further elongating it by the normal kind of pathway. So you end up with these monomethyl branched chain fatty acids with a single branch on the terminal end. And we found that when we fed in, say, different short chain fatty acids that could come from the microbiota, such as propanol-CoA or butyrate, that fatty acid synthase would also use these to make fatty acids. So what we were showing is that fatty acid synthase is actually more flexible than what was previously appreciated, and that the metabolic environment of the cell and, say, the short chain fatty acid environment, or also the CoA balance, which can be changed by amino acid breakdown and different pathways, can all feed into the fatty acid composition of the cell, so that the CoA balance drives the fatty acid composition. So we then wanted to know if they change with disease. We found that branched chain fatty acids are decreased in adipose tissue of mice on a high-fat diet, and they're actually the most differentially abundant fatty acids in this context. And then when we calculated synthesis, we see that synthesis is really significantly suppressed in obese adipose tissue. And then finally, we also looked at patients. So we worked with Rohit Lumba and UCSD, who's focused on non-alcoholic fatty liver disease, and we found that branched chain fatty acids are decreased in the blood of these patients that progress from NAFL to NASH, which is a more detrimental stage of the disease. So now, overall then, our branched amino acid work showed that branched amino acid catabolism is suppressed by obesity-induced hypoxia, and this might contribute to the elevated levels we're seeing in obese and type 2 diabetic patients. And that this decrease in catabolism can also lead to decreased branched chain fatty acid levels, and that this might have some functional consequences. So this is kind of one of the main questions that I hope to pursue now is, what is the functional role of branched chain fatty acids in the body? So they're well known in bacteria, where they modulate membrane fluidity, but really what they do in humans is relatively unknown, and we think they might kind of act as some kind of signal between the adipose tissue and the liver, because we have some evidence showing that they can activate PPAR alpha. And there's also a lot of evidence showing that increased de novo lipogenesis in the adipose tissue, which would increase branched chain fatty acids, always has kind of beneficial outcomes on the liver. And then in addition to that, where they're most well known in humans, other than kind of as products of adipose tissue, is in breast milk. So they're present at really high amounts in breast milk, and there's a really wide variety of them present in breast milk. And they're also really, really abundant in vernix, so the waxy substance that kind of covers babies when they're born, and in the meconium. And it's not known exactly what they do, but there is some evidence that feeding these can protect against necrotizing enterocolitis in newborns. And we did collaborate with Africa O'Sullivan here in the department and show that they are really abundant in breast milk, and others have shown this as well. But what we also saw is that they're completely absent from formula. So now another kind of question of the lab is, well, why are branched chain fatty acids present in breast milk? Do they help with lipid droplet formation? And also, what impact do they have on the infant? And so finally, just a quick acknowledgement on the main funders. So this has all actually been mainly, all the work had been mainly funded by private funding. So most of the branched amino acid work was funded by Adipose. They had a general call out, this Innovation Alliance program, when I started my postdoc, where they're giving grants for any amino acid focused work. And then we started working on some of the breast milk due to this Mother's Milk Center of Excellence, which was set up in UCSD by the Larson Rosenquist Foundation. And then finally, I did all the work in Christian Metallo's lab in UCSD, and there was a number of different grad students and undergrads that helped out along the way. And then finally, just really quickly, sorry, Jar, the main questions now are just, how does diet-induced alterations in lipid flux drive NAFLD progression? Explore novel products of adipose lipid synthesis and how they might be beneficial. And then also explore more why branching amino acid catabolism might contribute to either tissue function or dysfunction in obesity. And thanks.